Selvi Rani from CSIR, Center for Cellular and Molecular Biology, Hyderabad, India. I'll be presenting the novel mutation in beta-MYH7 gene and their role in cardiomyopathies. The morbidity and mortality rate of cardiovascular disease are very high in South Asia, particularly in India. India is known for its ethnic diversity, and it is a potential source for population-specific genetic variation. According to the World Health Organization, around 60 million people they suffer due to heart diseases. Around 2 million people that die due to heart failure every year. That accounts for 27% of the total death. Although number of lifestyle choices which contribute for the development of heart disease, genetics plays a major role. It needs an extensive study. See, when you consider the sudden cardiac death in all age group, it is mainly due to cardiomyopathies. Then what are cardiomyopathies? Cardiomyopathies are the disease of the heart muscle which changes the morphology of the heart. Based on the morphology, it is classified into hypertrophied and dilated cardiomyopathy. In the case of hypertrophy, the left ventricle gets thickened. Here you could see. And in the case of dilation, it's the dilation or enlargement of the left ventricle. Sometimes it happens for the other ventricle, right ventricle also. In this, because of the thickening, the filling efficiency of this left ventricle get affected. Because of this here dilation, because it's here it fills more, pumping efficiency get affected. So ultimately it affects the pumping efficiency. So it is a clinically and genetically heterogeneous variable with the penetrance. When you consider cardiac muscle, it is made up of bundles of myofibrils. It is a repeated units of sarcomere. The sarcomere genes are the major gene. Say this actin or myosin are the major one involved for the muscle contraction. The other regulatory sarcomeric proteins like tropomyosin, troponin complex, and myosin light chain are involved for the regulation of this contraction. I'll be talking more about this. I'll be talking on myosin and this mutation, how it is contribute to the cardiomyopathies. When you consider the mutation in sarcomeric gene, it is a known cause for cardiomyopathies. This R2Q mutation in beta-MYH7 gene was the first one identified in sarcomeric gene in a HCM patient. After that, hundreds of mutations have been identified. In this identified mutations, 75% of them are from myosin heavy chain and myosin binding protein C. Only 10% of mutations were identified in troponin T and troponin complex and tropomyosin. But in the case of actin and myosin light chain, very rare mutations were observed, that too in a large cohort study. This majority of disease-causing mutation, which we got it in our study, even the study which were reported, there are unique mutations, rare and private, that occurs in families. Though these causative mutations were validated previously through co-segregation and linkage analysis, yet the molecular mechanism for the causative mutations have not been understood fully. Unfortunately, not many sarcomeric studies, not many studies on sarcomeric genes are available in Indian population. So we have analyzed 101 hypertrophic cardiomyopathy cases and 147 dilated cases versus 207 healthy controls. We collected samples from 3 states from India. One is Hyderabad, another one is Tamil Nadu and Kerala. These are the hospitals which were participated in this study. Our study revealed, when we analyzed these many samples for beta-MYH7 heavy chain gene, using direct sequencing, we got 52 variations. Of this 52 variation, 36 were novel variations. And in this novel variation, 9 were amino acid substitutions, and 8 of them happened to be in the head motor domain, and 1 is in the tail domain. We found 3 prime shift mutation and 3 splice site variation. And these novel mutations we have deposited in the NCB database. This 8 amino acid substitutions in the head motor domain, we further analyzed. Why only this mutation? Because everyone knows that head motor domain is the one which binds to the active sites on actin and it regulates the muscle contraction. So this is the functional domain. So the mutation which were present in this motor domain, we analyzed further. We found that out of 8 mutations, except alanine to threonine at 423, other 7 variations, mutations were evolutionarily conserved across species and predicted pathogenic by Polfin 2 and SHIP bioinformatic tools. And this PDP structure is available. This is the myosin head motor domain 3D structure, which is available in PDP database. We downloaded from there. And these are the mutation, these are the place where loci where we found our mutation. These are the 8 mutation. And we blasted this one and found that except our mutation site, it shows 99% similarity. And we made homology models for each of this mutation using Swiss model system. And each of these 8 mutants were superimposed with a 3D native template. And then we calculated the root mean square deviation. And this 8 mutation, and these 3 showed more deviation. This one showed moderate deviation, and this 4 not shown much deviation. And then we studied the non-covalent interactions for 8 of this mutant versus native template. We all know that non-covalent interactions are the one which determines the 3D structure of a protein. It is heart of stabilizing the protein structure. So we wanted to know how this mutant is deviating the structure. So we compared each mutant with the native one. And then we again found these 3 showed disturbed a critical network of non-bonding interactions at the mutation site. So these 3 showed more mutation, critical network. So I wanted to explain it one by one. In this case, valine is mutated with methionine at 431 position. Both valine and methionine are non-polar residues. This methionine, when the mutant methionine, it forms one more extra hydrophobic interaction with leucine 352. And this interactions buries this molecule further inside the structure, further inside the hydrophobic core. Because of that, as a result, it reduces the spatial distance between the other hydrophobic interaction in this molecule that produces strain and deviation in the structure. So we understood this deviation is because of this mutation, which is producing the extra hydrophobic interaction in the hydrophobic core. That could be the reason for more deviation here. This mutation we observed in 2 woman candidate. One is HCM woman and another one is DCM woman. That HCM was 55 years old and DCM was 45 years. This DCM woman showed more severe disease phenotype. Then we understood the DCM woman carried allelic heterogeneity. That means it has extra mutation in the same locus. Then we analyzed this. We found this alanine to threonine, one mutation, and one splice site variation, and one synonymous mutation in DCM patient. But HCM patient carried only one, that is valine to methionine. Even this 45 years old woman, her daughter was found to be having mild DCM. She also carried this mutation, but she is asymptomatic. And the family history of even the sudden cardiac death was also observed in the family. Her sister, when she was 55, she had this heart failure. And this, therefore, the age of onset and severity of disease highly variable within the family itself. This may be the lifestyle or environmental factor which is playing here. This work I have presented in the 11th annual conference of Winker Association at Bangalore Medical College Research Institute, and I was awarded Selvi Women's Research Award for this. And then I have spoke about this variation. Then we'll see these three variations one by one. These three, arginine to histidine at 723 and phenylalanine to 5 and leucine and histidine to leucine. Here you see it is this mutation. This mutant is co-segregating along with the disease in the family, therefore showing strong association. Here in this mutation, arginine to histidine, also we observed in two cases. One is DCM. That person is 48 years old, and another one is 42 years old HCM patient. This DCM patient again showed allelic heterogeneity by carrying additional variation in the same locus. One is splice site variation, another one is synonymous variation. Here also in this histidine to leucine, here also we found one more synonymous mutation in the same locus. Let's see this arginine to histidine. Here, when we did the non-bonding interaction, when I saw that I made this homology model, I studied the non-bonding interaction for this mutant. I understood that this histidine, what happens, this histidine forms one more hydrophobic interaction with the proline 727. This hydrophobic interaction pulls this molecule towards this one, and even this proline 727 again forms a hydrogen interaction with the proline 731. That pulls this molecule towards this. Because of that, the electrostatic interaction between these two molecules get broken down, and even the hydrogen bond here got broken down. This is the native one. See the structure, how it is getting disturbed here. Not only that, because this molecule, it pulls this one towards this one. Here, the hydrophobic interaction between two isoleucine also got, this is the bond here, original bond, gone destroyed. So it is deviating structure like this. And this proline, we know, the proline residue is a unique one. It lacks the amide proton, and that it cannot donate hydrogen to stabilize other bonds and promote stability. As a result, it is deviated more here. The next mutation, phenylalanine to leucine. Here, this phenylalanine is replaced with leucine. This leucine is forming hydrophobic interaction with the tryptophan here. This bond interaction pulls the leucine towards the tryptophan. As a result, this hydrophobic interactions here, B and C, you could see here, could not be existing here because it is pulling the molecule towards the tryptophan side. This bond got broken down, and this is deviating. This got deviated this side. Because it is got deviated this side, it forms an hydrophobic bond with the lysine 766. But the thing is, lysine is a crucial molecule here because this myosin head, this is the head which carries the active site for actin, goes and bind to this one. This molecule is happened to be in the ligand binding site. So the contraction itself get disturbed here. That could be the reason for more deviation here. When you consider this E to K, not much deviation is here expecting, but the patient is severely affected. What could be the reason then? Then we found that patient is carrying compound mutation. We found that this patient carried one more mutation in the tropomyosin gene, that is a regulatory gene, and actin gene, one more mutation. And in this myosin itself, in the tail domain, one more mutation is existing. That is, the severity of the patient is because of the compound heterozygosity. We even studied the distance relationship contact plot, mutant versus native. Here because we wanted to know how a single amino acid substitution could find on the whole protein, mainly the difference in distance between the C alpha, C beta, and side chain amino acid pairs. We studied the unique one in mutant, the unique one in the native, and the common one between them. The amino acid pairs between the C alpha distance, the C beta distance between the amino acid pairs, the native one, the mutant one, the common one. Again, the side chain interactions between the mutant one, native one, and the common ones. Then we calculated the chi-square deviation. This is the C alpha amino acid pairs and C beta amino acid pairs for the distance contact plot. And then this is the side chain amino acid plot. And then we even calculated that interactions relationship contact plot. That is the main hydrogen bond interactions and residual type interaction. In the case of hydrogen bond interactions, we studied the main chain interactions, main chain side chain interaction, side chain interaction, and multiple hydrogen bond existing within the atom we studied. And also I studied this residual type interactions, hydrophobic interactions, and hydrophilic interaction. I studied the hydrophilic and acid-based interaction. These are the plot which we studied. Even we studied the Ramachandran plot to visualize energetically allowed region for backbone dihedral angle between the phi and psi in mutant versus native structure. We all know that the genetic information that denotes the fold contains in the amino acid sequence when hydrophobic residue that aggregates in solution to form a well-packed nuclear core. When it forms a nuclear core, not only the hydrophobic residues forms a nuclear core, it along with that some peptide groups and polar side chains and also charged side chains will also get buried in the interior molecule. That also plays a key role in maintaining the protein 3D conformation. The protein structure determines the function of the molecule. If it loses its shape, no longer it is functional. Abnormal protein itself is pathogenic agent for many diseases like Alzheimer's, Parkinson, prion disease, and type 2 diabetic, etc. This we were wondering how a single amino acid can make it. It is not only now, it is previously it is reported in the sickle cell anemia, glutamic acid at the 6th position valine is replaced. It increases the hydrophobicity. This RBC sticks to each other in order to avoid the aqueous environment to alter the RBC structure from round shape to sickle shape. Here also we found a few mutations uniquely disturbing a critical network of non-bonding interactions and deviating the structure more. And also we found that allelic heterogeneity could be the contributing factor for the DCM phenotype and its phenotypic plasticity. The study profoundly increased our understanding about the mutant frequency in our population and its impact. It is useful for the personalized medicine in order to block or diminish the disease phenotype, particularly each mutant can be treated specifically. We all wonder that DCM and SCM are entirely of two different phenotypes. How a single mutation can give rise to two different phenotypes. Like in the mouse, like heterozygous 4, the first mutation R2Q, it was reported in HCM phenotype. When they crossed this heterozygous R2Q, they found the homozygous mouse giving a DCM phenotype. That means, is it when it is more severe, is it a HCM? When it is uncompensated, we leave it like that, it becomes DCM, or a two distinct pathway or gradation of a single pathway. The animal model, a single heterozygous mutation can give rise to HCM and DCM in probably due to the impact of modifying factor. Or HCM and DCM are two different mutation and two different pathophysiology mechanism. Therefore, it is different views is existing. But therefore, the mechanism that causes HCM and DCM are still under speculation. But our study shows some light towards it and increase our understanding about the mutation frequency, how it can be treated in future. So this will be very useful for the personalized medicine. That's all. From here, I finish my work presentation. Thank you so much. First of all, I thank the organizer for giving me the opportunity to present my research work in this conference. Thank you so much.
Deepa, for your valuable presentation.
Yeah.